Start. Hello, good afternoon to all. I am Dr. P. Rais Aziz. I have done masters in the subject of veterinary medicine from Rajwas Bikaner. I am assistant professor in the School of Veterinary Sciences, Abhilashi University. So let's start. The topic of my presentation is foot and mouth disease. The synonyms of the disease are aphthous fever, contagious aphtha, benzotic aphtha, aphthosa, fast-moving disease, and tiger heart disease. Now, the epidemiology and transmission, we will be discussing about the transmission and the epidemiology of the disease. The ways of disease transmission are infected animals. From the infected animals, it gets spread to the other healthy animals. And second is the wind, through which there is transmission through the air to the various uninfected areas of the animals. And next is the garbage and disposal of the farms. And second way of transmission is animal origin products like the feces, the milk, the urine, and other things which are derived from the animals likewise. And next is the feed and water. From the feed and water, it also gets transmitted from the unhealthy, from the infected animals to the unhealthy to the other animals. And likewise the people, from the people which are currently working, in the farms, from these animals, from the people, it gets spread to the healthy animals. Next is the vehicles, the trucks which we use in the farms. From the trucks, from the vehicles, on the tires, the garbage, the disposal items get attached and they get transported from the infected to the uninfected animals. And likewise the clothings of the people and the shoes of the people, they also transmit the disease. And bedding of the animal which we use inside the farms, they also get contaminated and hence affect the healthy animals. And from last is the infected, from the infected animals to the non-infected animals. These are the ways of transmission through which the FMD disease gets transmitted. Next is the definition. The foot and mouth disease, it's a highly transmissible disease which is caused by the infection with a family of the virus Picornaviridae and genus Aphthovirus. There are many other genuses of under which come under the Picornaviridae, which cause diseases in animals like Aphthovirus is there. Second is cardiovirus, enterovirus, arbovirus, humnivirus, cubivirus, and many other diseases, other genuses which cause diseases in the livestock animals. The Picornaviridae, it has a specific characteristic. It is the smallest known virus of animal origin, which is the smallest known. So there are the 7 serotypes of the aphthovirus Picornaviridae, which causes diseases, which cause the disease FMD in the animals. Like the 7 serotypes, which we will be discussing, like the 7 serotypes are AOC, Asia1, and SAT123. The name of these serotypes come from the various places of origin from which they were first originated. Like A and O came from the France, C in Germany, Asia1 in Pakistan, and SAT123 in many South African territories. So these are the serotypes which cause the FMD disease. Next is further diversity is found because each of the 7 serotypes, they are different from one another. There is further diversities between the serotypes. So this is the reason why the vaccination is not successful in the whole world, due to the diversity between each serotype. There is the diversity between the serotypes, and the virus affects mainly cloven-footed animals of the order Artiodactylia. Likewise, the livestock hosts which are affected from the disease are cattle, pigs, sheep, and goats. Many other wildlife are also affected, like the deer, antelope, or African buffalo, giraffe. But it mainly affects the Artiodactylia order. Now the next is epidemiology and transmission. The virus which we are talking about, the picornavirus or the genus aphthovirus, it is resistant to many external things like many disinfectants, many meat storage practices which we currently use in our meat storage technology institutes or feed technology institutes. Likewise, the virus of the FMD disease, the picorna or the apthovirus, it is remaining viable. It remains alive in many infected premises for more than a year, and in clothing and feeds, it remains viable in for about 10 to 12 weeks, and in hay and bran, it remains viable for 4 to 5 months, and in the lymph nodes for 3 to 4 months, and in the bone marrow, and 1 month on the hairs of the animals. And in infected, if there is infected frozen bull semen, it also remains viable. And the virus, it's killed after boiling for 15 minutes, but we have to depend on the protocol, on the thing which we are using for boiling. Like if we are pasteurizing the milk, then it's good, but pasteurization also, if we are using the correct method and for the correct time, then only the virus can get killed. Next, it is resistant to alcohol, ether, and chloroform. It is not getting killed by these of the things. It is destroyed by 1 to 2% sodium hydroxide or formalin and 4% sodium carbonate solution. We are talking about the virus, how it can be destroyed or how it cannot be destroyed. Next, it is generally found in the form of outbreaks. Whenever an farm is affected, one animal is affected, from that one infected animal, it gets spread to other animals also. And from the one farm, it gets transmitted to other farms. So the disease, which is generally found in the form of outbreaks, and it spreads from one herd to another very fast, it gets spread. And if we are talking about our country, India, the southern region of the country, the southern states experience the most highest proportion of the outbreaks, which is followed by the eastern states and then the northeastern states. The proportion is shown, for example, the southern region gets affected, 40% of the outbreaks are there. Out of the 100%, 20.5% is found in the eastern region, and in northeastern, it is 18.3%. But in the northern, western, and central regions of the our country, it is only the proportion of outbreak is only 6 to 8%. Now we will talk about the morbidity rate. Morbidity means the infection which we are having, a symptom of the disease or the infection, or the amount of disease which is present within a population. That is known as morbidity. So the morbidity during outbreak, it reaches 100%. That means whenever there is outbreak, it affects all the animals of the farm. But the case fatality or the mortality, the death rate, it means the death rate, the number of deaths which are present at a specific time, the death rate is very low, which is about 2% in adults, in adult animals. But in case of young cows or young piglets, the mortality rate is 20%. That means the young cows and young piglets are very affected by the disease, in which there is the malignant form of the disease. It affects the heart, it causes myocarditis. Due to the myocarditis, the animals, the young cows and the young piglets die. So the source of infection through which the infection gets from one animal to the other is the infected animals, the carriers, the infected materials, the meat, infected meat is there, the semen in which there the virus is viable, and other animal products. These things transport the virus from one of the from one animal to another animal. The virus is also present in the secretions of the animals, like the milk, the saliva, the urine, feces, and semen. And the transfer, it also occurs from one animal to another animal. It occurs by direct contact. Whenever an animal is in direct contact with another animal, so it gets transferred. The main portals from through which the it gets entry into the body, it is the inhalation and the ingestion of the virus. Next is the pigs, which contract the disease by ingestion of the infected meat scraps, and spread from these pigs to cattle occurs by movement of people, abattoir waste, or animals. The pigs are the disseminating thing which disseminate the virus very quickly. So from pigs, the virus gets very quickly disseminated to other healthy cattle or other healthy pigs. Now the airborne infection through the air it gets spread from one place to another place. The virus gets spread as far as 250 kilometers. It has been seen that the virus can transfer through wind currents, particularly whenever the atmosphere is humid, there is humidity, so the virus can transfer from one place to far off places. Now, the symptoms, the characteristics which we can see in the FMD, whenever an animal is affected by FMD, there is fever. Fever is there, and the vesicles, the blisters appear inside the mouth, on the muzzle, on the teeth, and on the feet of the animal. These are the clinical signs which we can see whenever the animal is affected. Besides, there is salivation, petylism is there, extra salivation, lacrimation, and other clinical signs are also there. The virus which we are talking about, the aphthovirus, it is transmitted via aerosol or respiratory secretions through the milk, semen, which we have already discussed. And in many species, susceptible population, morbidity reaches 100%, but with rare fatalities. In the young animals it causes fatalities, but in adults it doesn't cause any such, because 2% is the death rate in case of adults. The FMD is distributed worldwide. It has been seen in all the parts of the world. But now, in the 21st century, the many countries from many countries it has been eradicated, like the North America and Europe. In the endemic countries, endemic means from a disease outbreak when it is consistently present. That is known as endemic. But whenever there is endemic, it is limited to a particular region. In endemic countries, like where the virus is continuously present, like the Africa, the Middle East, and the Southern Asia, it places economic constraints on the livestock trade, because whenever there is livestock trade is there, but the countries which are affected, so the trade barrier is there, and it can be easily reintroduced into the disease free animals. Unless strict precautions, we will not place precautions, then the disease gets easily spread from one place to another place. And outbreaks, whenever there is the outbreak of the disease, it severely affects the livestock production in terms of milk production. Milk production is severely disrupted whenever there is outbreak of the FMD, and it requires significant resources to control. Whenever we have to go for significant control of the disease, for vaccination is there to control, and whenever there is the FMD, then we have to go for the treatment. But when if we see this is a viral disease, so viral disease, it can't be treated as such, but accordingly we have to give the symptomatic treatments. And in non-endemic, a major part is lost, major part of the cost is lost because of the lost trade. Because if there is the outbreak, there is the trade is lost. So a large number of animals we have to cull the animals in order to limit the spread of the disease. Further, we have to limit the spread. The different serotypes which we already speak, the seven serotypes, these are not uniformly distributed all over the world. Because if we see 70% of the outbreaks which occur globally all over the world, only serotype O is responsible for 70% of the outbreaks, and other six serotypes, they only uh, cause 30% of the outbreaks. And among the seven serotypes in Africa, only six have been seen among the seven serotypes, like O, A, C, SAT1, SAT2, and SAT3. And in Asia, only four serotypes we have been isolated, and in South America, three serotypes we have isolated. And since 1995, serotype C has not been reported in India. And the serotype C, before 1995, it was the most common and the dominant type in India, followed by the serotypes Asia 1 and serotype A. Between the 10 years of 2011 and 2020, a total of 2,226 FMD outbreaks were recorded. We are talking about the number of outbreaks. We are not talking about the number of animals which were affected by the disease. So the number of animals was in millions, in lakhs. But the outbreak, because if one outbreak is there, thousands or hundreds of animals are affected. So the number of outbreaks was total of 2,226. And during the years 2013 and 18, our country recorded the highest number of FMD outbreaks in during these 2013 and 2018 during these years. But in 2020, during the year 2020, it had the least number of outbreaks. Now many countries are free from the FMD, like North and Central America, Australia, New Zealand, Greenland is there, Iceland, and Europe. And in the Europe, it was in Bulgaria in 2011 that the last outbreak was recorded. But vaccination against the serotype C still occurs in some places, because the complete absence, if we see the complete absence of the virus, is difficult to prove, because all the outbreaks, if there are outbreaks or the large number of animals are affected, the all the outbreaks are not recorded by the government. So complete absence of any of the serotype is still not possible, and it's difficult to prove whether the serotype C or whether the serotype O is present or absent in the field conditions. The FMD, which we are talking about, the virus, it's transmitted directly with the infected animals, or direct contact with the secretions or excretions of the animals, or by mechanical vectors. The carrier vectors are there, for example, the people or the animals or the birds, vehicles, or air movement over land or water, because it gets transmitted through the air, over the land, or on the water. The virus which enters the host animal, it enters through inhalation or ingestion or the skin wounds and the mucous membrane of the animals. But the important point is the South African territory virus, the serotype South African territory, it is venereal also in African buffalo populations. Through venereal route, it also gets transmitted. You can see in the epidemiology a potential scenario for introduction of the virus into previously FMD areas for a susceptible population, such as pigs, to give given imported food derived from an infected animal. Whenever there is an imported animal, we give him some imported food, there is also the chances of the spread of FMD virus. It also spreads from pigs, because I also told you already that the pigs are the disseminating virus. They can expire 3000 times more virus than cattle, and these viruses then cause infection in the susceptible cattle hosts via the aerosol route of transmission. So the virus which was reported, once it was reported to travel over water more than 150 miles. It has been reported that it has traveled over more than 150 miles from Brittany, France to the Isle of Wight, UK in 1981. But over land, it travels no more than 10 kilometers, because on the water there is the humidity. Humidity is there, that's why it travels more in on the water as compared to the land. And if we talk about the agro-terrorism potential, FMD has got the potential because of the high rate of the infectivity by which it gets infected to the animals, and the high transmissibility through the wind and by the fomites. It causes huge losses to the animals in terms of milk production or other things. People acts as the mechanical vectors of the virus, because the virus gets attached to the clothing or the skin of the animals. But the thing to note here is the FMD, the foot and mouth disease, is not a public health, it's not a zoonotic disease, although it is environmentally resistant, but still it can be inactivated at a pH less than 6. Whenever there is pH less than 6, it can be inactivated, or greater than 9 pH is there, it can be inactivated. And by desiccation and at temperatures, whenever there is temperature greater than 56 degrees centigrade, the FMD virus, the apthovirus, the picornaviridae, it can be inactivated. Next is it is resistant to lipids like ethers, solvents, but in case of sodium hydroxide or sodium carbonate, citric acid, and acetic acid, these are the effective disinfectants which we can use on the surfaces in order to disinfect the surface from the FMD virus. Likewise, less effective disinfectants are iodophores, quaternary ammonium compounds, hypochlorites, and phenols. FMD virus, which we are talking, is shed into the milk in dairy cows before the clinical signs develop. Whenever an animal is affected, it is shed into the milk. the milk is getting infected before the clinical signs develop. So the virus which we are talking is spread from farm to farm. uh, it gets an opportunity to spread, and from cow to calf it gets spread. It gets transferred through the raw milk, because before the clinical signs develops, the infection is there and it gets transmitted into the milk, and it infects the calves also. And as we have already shown you, the mortality rate is 20% in case of young calves. And it also may survive pasteurization depending on the method which we are using, because on the in the pasteurization we use the time and the method which we are using, how much time we are giving to the method, and for how long we are pasteurizing the milk. So the pasteurization method is also important, as it may survive it. And the FMD virus, it survives on many things for many years, for many weeks. Likewise, on hay and straw bedding, it can survive for 20 weeks. In dry fecal, it can survive for 40 days. In summer, in winter, it can survive for 6 months. In the fecal slurry, in urine, it can survive for 39 days. And in soil, in case of summer season, it can survive for 3 days. And in winter, in the soil, it can survive for 28 days. But it depends on the initial level of contamination. What was the initial level of contamination? So these are the time periods in which the virus is viable, and it remains in the different things, it remains viable. Now the virus characteristics, we got the virus characteristics. Likewise, we know it is a one of the picornavirus family is there. It is a positive sense RNA. Positive sense means these are a group that are single stranded. Positive sense RNA genome is single stranded, made of RNA, ribonucleic acid. The RNA sequence which we are talking about, the virus, it is 8500 nucleotides in length, and it includes a single large opal reading frame that encodes a large polyprotein which is more than 2330 amino acids in length. Open reading frame, if we are there comes the word open reading frame, it means it is defined as spans of DNA sequence between start and stop codons. So during and after the synthesis of this polyprotein, it is processed largely by virus encoded proteases to generate 15 mature proteins are generated, and the structural proteins of the virus are only four. It is VP1, 2 and 3, which are on the outside of the virus, outside of the virus envelope, and VP4, which is present inside the virus. So these are the proteins. It is mainly 15 mature proteins. One large polyprotein is there, and these generates 15 mature proteins. And main the structural proteins are only four, as you can see from the photograph. The virus capsid, it serves, it protects the host cell from getting entry into the cells. It binds to the specific receptors on the cell surface. After virus internalization, whenever the virus gets inside the body, inside the cell, the RNA genome which is present, because it's a RNA type of the virus, it is released into the cytoplasm of the cell, and then it is translated to make the viral proteins, including non-structural proteins, and then the RNA genome which gets replicated. Many thousands virus particles are produced inside a infected cell within a few hours. So there's the viral multiplicity of the virus. Within a few hours, it gets multiplied. Now the pathogenesis through which the disease gets spread from the virus gets infected, it gets entry into the animal, then it gets entry into the respiratory tract of the affected animal, and main replication of the virus occurs in the pharynx and the epithelium of the mouth, epithelium of feet, and on the teats. Inside the teats, it gets main replication occurs there. And then whenever there is replication, there is multiplication of the virus. From the multiplication, it gets into the blood. There is viremia inside the infected animal, and whenever it gets inside the blood, viremia, then it gets it comes outside of the body from the like saliva, from the semen, feces, milk, nasal discharge, and urine. These are the routes through which it gets spread into the environment and affects the other healthy animals. The primary site which we are talking about, the replication, is the mucosa of the pharynx. From the pharynx, it is the primary site of infection and the replication of the virus. And then the virus can get entry into the skin lesions or through the gastrointestinal tract. It can also get uh, entered into the body. And once it gets entered, it affects the lymphatic system, and then it replicates in the mouth, muzzle, teeth, feet, and areas of the damaged skin, which gets affected in pigs, like the knees and the hock joint of the pigs. And then whenever the virus gets entry, it then develops vesicles, get developed inside, mainly inside the mouth cavity, and then the vesicles rupture within 48 hours. And when there is the recovery of the animals, the recovered animals, they become carriers. And whenever there is vaccination, we do vaccination, then also the animal become carriers. But whenever the animal is a carrier, it has a low level of infection, so it doesn't pose such an threat to the other healthy cattle, because it is a carrier. And whenever animal is a carrier, it has a low level of infection rate. Whenever animal becomes carrier, the carrier state, which lasts for 3.5 years in case of cattle, and 9 months in sheep, and greater than 5 years in African buffalo. But whenever a pig gets infected, it doesn't become a carrier. This is the most important fact, that pig is not a carrier, but it is a disseminator of the virus, and it causes more infection as compared to cattle in case of disseminating the virus. The risk posed by these is low, because it is not possible under controlled conditions to transmit the virus from the carrier cattle to the naive cattle by close contact for extended periods. So this is not a threat, because the carrier cattle doesn't pose such a threat. But transmission of this virus has been achieved from carrier buffalo to cattle. This is also one of the important point. And now the incubation period, it depends on the host, which is the host, and the incubation period is different for the different hosts. Like the incubation period in case of sheep and goats is 3 to 8 days. And it also depends on the environment, because if there is humid environment, then it gets spread quickly. And the route of exposure, what was the route? It was through inhalation, or it was through the wound in the GIT, or any other route. And the virus strain, which we are talking about, the 7 serotypes, what type of serotype is present in the animal. So the incubation period, it depends on these four things. And in case of pigs, the incubation may be greater than 2 days, and in case of cattle, it can be from 2 to 14 days. In case of cattle, the incubation period is there. But the incubation period can be short as 18 hours for many pigs under intense direct control. It is important to be aware that animals can transmit the virus before, as we are already told you, animals can transmit the virus before the appearance of clinical signs, because the virus is present in the pharynx, and it comes outside of the body through the excretion, through the secretions, and before the appearance of clinical signs. Now, the clinical signs is the occurrence of the blisters or the vesicles inside the mouth cavity. Foamy salivation is there. Blisters appear on the teeth and on the feet also. These are the main clinical signs. Excessive salivation, petylism is there, slobbering, we can say. Lesions are present on the coronary band. Lesions are present on the tongue. Their vesicle formation, ruptured vesicles are there. And on the mammary gland, you can see the clinical signs. The lesions are present, the blisters are present. Besides these clinical signs, other clinical signs are present, like the laminitis is there, because it affects the feet of the animal. Reduced milk production is there. Anorexia is there. Abortion may occur. Sudden death in case of small animals like small piglets and small cows. Fever is there, and weakness is there. Now the clinical sign, as we have already taught about, there is the fever, which may be greater than 40 degrees centigrade. The vesicular lesions occur on the tongue, the hard plate, inside the mouth cavity. You can see all inside the mouth cavity there is the presence of the lesions. And there is excessive salivation, petylism is there. Stamp their feet, this animals stamp their feet, they put their feet hard on the ground, because the lesions are present on the feet, and the animal prefers to lie down, because whenever animal stands, it causes pain because of the lesions present on the feet. The oral lesions, they coalesce, they combine and form erosions. But after 11 days, vesicle formation is there. And vesicles on the feet, they take more time to heal because of the bacterial infection, and it may lead sometimes to chronic lameness. And secondary bacterial mastitis also occurs, because whenever there are vesicles present on the teats, the animal is resistant to milking, so mastitis also occurs. And whenever the vascular disease develops, animals lose condition, they become very cachexic, they lose weight, and milk production diminishes. And in case of young cows, there is the myocarditis, and due to the myocarditis, the young cows and young piglets die, because 20% is the mortality rate. The infected pigs show lameness, and blanching means skin discoloration is there around the coronary band, and they develop fever of 41.5 degrees centigrade. And the affected pigs become lethargic, they huddle around other pigs, and they don't eat their feet. Vesicles develop on the coronary band, the heel of the foot, including the accessory digits, the snout, mandible, and the tongue of the pigs. Additional vesicles may also form on the hock and knee of the pigs, which are housed on rough surfaces. And depending on the severity, the horn of the foot, they may slug off and may cause chronic lameness in the recovered pigs. And pigs less than 14 weeks of age may die because of the myocarditis. And this is more frequent, the myocarditis is more frequent in pigs as compared to cows. And in case of sheep and goat, the clinical signs are largely inapparent. The first clinical sign which we can see is the lameness in the animal, and after the lameness, there is fever, there is vesicle development on the interdigital cleft, the heel bulbs, the coronary band, and the mouth. Vesicles also form on the teats of the female animals, and rarely on the vulva and the prepuce of male. And secondary infection also occurs because of the reduced milk, the chronic lameness, like other viral infections also occur. And in young pigs, they die. And young sheep and goat, they also die without showing any clinical signs due to the myocarditis or due to heart failure. If in case of camels, we have seen that experimentally infected animals are there, and they have mild clinical illness, but severe infections can happen in those camels, like salivation, mouth lesions, and slogging of the footpad and skin of the tarsal and carpal joints. Water buffalo likewise have mouth and foot lesions, but they heal faster, and less severe lesions are present in the water buffalo as compared to the cattle. Likewise, FMD also gets infected in the wildlife animals, but in the wildlife there is slowing of the antlers in case of deer, or toe horn they have been reported in the case of wild animals. If we talk about the gross pathology, there are the vesicles, erosions, and ulcerations inside the mouth, and on the coronary band, on the heel of the digits, there is several ulceration and erosions, and on the mammary gland, teat, and prepuce, you can see the erosion and ulceration. And there in case of young animals, they show yellowish to grayish bands of myocardial necrosis, and which gives the characteristic name of the tiger heart appearance in case of the younger animals. In adult cattle, like pericarditis occurs with pinpoint hemorrhage on the myocardium. The lungs show grayish and firm patches on the surface, but there is hepatomegaly in case of, and it is congested in case of FMD. Whenever there is FMD, the spleen gets enlarged. Splenomegaly is there with multifocal irregular grayish white necrotic areas occur, and on the ruminal pillars show hemorrhagic and eroded areas. Besides, the rumen also shows the clear ulcers also. Next is this diagnosis, how we are going to diagnose the disease. There are many diagnostic kits which are available, like RT-PCR is there, virus isolation is there, antigen detection ELISA is there, uh, CFT, complement fixation test, virus neutralization test, solid phase competition, enzyme linked immunosorbent assay, or liquid phase blocking ELISA is there. Now the differential diagnosis, how we are going to difference between the diseases, because many diseases are there which are showing the same clinical signs. Like in case of cattle and pigs, vesicular stomatitis disease is there, which is caused by the Rhabdoviridae family, and genus is vesiculovirus. And likewise in pigs, swine vesicular disease is there, which is caused by enterovirus of the picornaviridae family. Vesicular exanthema is there, which is caused by calcivirus, and Seneca valley virus, which is caused by the picornavirus. So these are the differential diagnosis. Other diseases which show same clinical signs, and it is indistinguishable. We cannot judge from the clinical signs whether these disease are present or FMD is present. But for this we have to go for the laboratory diagnosis, and for the laboratory diagnosis it comes under the containment group 4 pathogen, because it is more pathogenic as compared to other organisms. It comes under this group. So laboratory diagnosis is essential, and whenever there is not any reference laboratory or regional laboratory, then OIE FMD reference laboratory is present there. So we have to put the samples and send them to the laboratory. For example, in India it is likewise in ICR Mukteshwar Nainital, it is project directorate on FMD is there present. So whenever if there is not any regional laboratory, we have to send samples on the reference laboratories. And the tissue of sampling, the choice is vesicular epithelium or the fluid which is present inside the vesicles. At least 1 gram of epithelium should be transported with the pH buffer 7.2 to 7.6, and the samples they should be refrigerated and transported on ice, and then virus isolation or reverse transcription assay should be done. Blood samples can also be tested, but viremia is short-lived, so whenever the lesions are healing, it has been cleared from the blood and antigens can be detected. Repeated sampling from the oropharyngeal fluid can also be done, but virus presence in such animals is always low in case of carriers, and it fluctuates inside the carrier animals. Now the treatment is part is there in regions that are normally FMD free. Control is typically attended by culling of the animals, because culling is the best method in order to control the FMD to spread, and animal movement we have to control the movement of the animals. In both normally FMD free and endemic areas, vaccination should be done, because vaccination is only the method through which we can control the disease. Likewise, the government of India, they have launched an FMD control program in 2003-4, which covers 54 districts they covered, and under the scheme NADCP in 2019, they covered the whole of the country, the National Animal Disease Control Program. It aims to control the FMD by 2025 by vaccination, and their aim is also to eradicate the disease by 2030. And no treatments are available, only symptomatic treatment we can give to the animal. And the vaccination, it gives protection uh, only for 4 to 6 months, so we have to give repeated vaccination. Likewise in India we have to give twice the vaccination to the animals. And other things are likewise like other disease, it has influenced development of international regulations by prohibiting importation, so the disease exists, so we have to kept in mind these important things. And vaccination, we have already told you, it gives protection for 4 to 6 months only. And in current vaccines which we are using, they are inactivated vaccines which contain adjuvants like aluminum hydroxide, saponin or oil. These are the adjuvants which we are using. And in the countries when the disease is endemic, vaccination is only the thing which we can use for the control. And two types of vaccination programs are available. First is the systemic vaccination. Here all the animals in the herd in the country, they should be vaccinated, revaccinated every 4 to 6 months. And second is the ring vaccination. This type of vaccination, it is practiced whenever there is outbreak, and we do vaccination in the vicinity, in the nearby areas, then we vaccinate inside, we go inside the area and vaccinate. The purpose of this vaccination, it is to create a barrier of the immune stock around the infected area, so the vaccination, so the disease is not getting spread to other areas also. And now the second thing is the precautionary measures which we have to use, so the disease doesn't spread, so the disease remains inside the area. So the prevention measures, it fall into three categories. First is the biosecurity measures for animals, like animal products, vehicles, people and the equipments. And next is the restrict or stop all the animal movements And next is observe, detect, and report any diseases or the unusual signs if we see inside the farm on the animals. So we have to contact our veterinarian, so if there is presence of the disease. Next step is many steps we can use, like on the farm entrance we have to limit access to your farm. The farm entrance is major control point through which we can control the disease. On the farms we should have only one gated entrance in order to better control and monitor the vehicles and the visitors. Keep the gate locked. When not in use, stop all movement of animals on and off of the farm. Whenever FMD is confirmed, movement restrictions should be implemented locally, regionally, and possibly nationally, so as to restrict the movement of the animals. Restrictions will depend on the scope, how much it has spread, and these measures will minimize the spread of FMD to other areas, including your farm. And post some signs on the farm, outside of the farm, in order to inform the visitors. Stay off the farm unless permission is given. Honk, you have to honk before entering into a farm, so to announce your arrival. Check with the farm personnel upon arrival. And next is the farm biosecurity procedures. What security procedures we have to follow? Likewise, wear protective clothing we have to wear while on the farm, while we are inside the farm. The visitors and their vehicles, they should avoid contact with the animals, but if it is necessary, then only they can contact with the, should have contact with the animals. Traffic inside the vehicle, on and off of the farm, should be closely monitored and it should be recorded. And we have to maintain a log sheet so as to record all the visitors and the vehicles that enter your farm. So the visitor should be accompanied from someone who works on the farm at all times. He should accompany the visitor. Accurate record keeping, we have to keep recording, surveillance and tracking, and we have to recall. So the ability to recall visitors and vehicles that were on the farm. Besides the vehicle, employees, limit employees to only those necessary which are necessary for the operation. Minimize contact, we have to minimize contact with the animals. We have to implement some biosecurity measures for the employees which are coming inside the farm. We have to give them clean boots, hats, and coveralls to wear on the farm. These items should remain on the farm, and whenever a person leaves on the farm, the items like the boots, hats, and coveralls, they should be washed and disinfected. And the footwear should be disinfected before entering. Hands must be washed with soap and warm water. And the employees, whenever they have contact, they should use biosecurity measures, like they have to educate. First we have to educate you ourselves and then the employees about the FMD, the signs, uh, like the blistering. If there is blistering or ulcer on the mouth, tongue, feet, or teats of the animal, then we have to contact some veterinarian. Or increased salivation, refusal to eat, animal doesn't eat, or signs of lameness, or it refuses to walk or move, then we have to contact our veterinarian. And about the neighbors, we have to discuss the threat of FMD with the neighbors, which are the farm neighbors. And we have to determine the steps which we can take together to protect the farm and the area of the farm. We have to eliminate direct contact between animals. Control, we have to control the roaming animals like the dogs and cats. We don't have to share the equipments on the vehicles between one farm or another farm. Always we have to wear the clean coats, coverall, boots, hats. When we come in contact with the animals, we have to change our clothes, our gloves, hats, and wash and disinfect them. And we have to wash our hands thoroughly whenever we come with contact with an animal. And then visitors, we have to put some signs. We have to minimize the visitors. It's a contagious disease, it gets attached on the people's clothing. So the virus can be carried. The virus has to be stopped by these things. The visitors should park their vehicle at the entrance of the farm. On only the vehicles should be used which are already using inside the farm, which don't go outside the farm. Those vehicles should be used. And biosecurity measures we have to follow. And for visitors, we have to provide the coveralls and boots and healthy clothings and footwear. And for the disinfection of the footwear, we have to use some brushes, hoses, and water washers. We have to use pressure washers. We have to use. All visitors, whenever there is visitors, someone from the farm who is working in the farm should accompany them. And livestock should avoid visitors. We have avoid livestock areas, pens, and barns. Whenever there is not necessary, they have to they should avoid visiting these places. And we have to visit at least away from affected areas for at least 5 days. Visitors should not be allowed access, and they have to follow all the above protocols. And vehicles, whenever there is vehicle, it is highly transmissible from the vehicle, it gets infected to the other farm. So the vehicles, we have to check, we have to wash them whenever they come inside the farm and they go outside of the farm. And these are the other things which we can use. Besides, pay attention to the tires, and other record keeping should be done in order to record the disease, the health status we have to record. And the livestock, don't allow with the other livestock, provide them fresh water and drinking water, fence off the streams and lakes. uh, Isolation should be done, because if there is not isolation, then animal can get affected, all the animals can get affected. And whenever there are the clinical signs, you have to contact the herd veterinarians. And we have to use these other things also, like separate facilities, equipments are there. First we have to see the healthy cattle, then at last we have to see the diseased cattle. We have to disinfect all the things, all the equipments. And we don't acquire the semen from FMD areas. And these are the quarantine measures we have to use. We have to isolate the animal, because from the isolation it can be prevented. Wildlife, entry of the wildlife should be minimized inside the farm. So the wildlife is also a carrier of the disease. Cleaning and disinfection of the things we have to done, the cleaning and of the vehicles, the boots and the buildings, the proper disinfection should be done. Proper clothing and footwear for the visitors, we have to use hand washes. We have to remove the clothing, and whenever we give visitors some clothing, they should be left inside the farm. Barns and buildings, they we have to uh, using hot water and detergent, we have to disinfect all the and buildings, and dispose the beddings and manure of the inside the farm. And vehicles, where they are potential tires and wheel wells, they are also important things which can transmit. So always use pressure washers, brushes, hoses, and other approved disinfectants for the tires of the vehicles and for the wheel wells. Last is the, you can see on the vesicles are present on the teats of the female animals. On the coronary band of the feet, you can see the vesicles, the blisters on the muzzle of the animal. And inside the mouth cavity, on the tongue, and on the gingiva, on the gum of the animal, on the oral mucosa of the animal. And last is the rumen mucosa, there is on the pillar of the rumen mucosa. Okay, thank you.